I think I announced it way too quickly. There was nobody there. It was fine. I'll try again.
I don't think people, I think the announcement should be like, we are now meeting.
Well, it's after 1:30. Yeah, we were giving it a little extra time. Hey, everyone. Before we get started with our next keynote, I had a couple announcements about tonight. So again, the cruise tonight, boarding starts at 7 p.m. We've gotten a lot of questions about where you actually need to go for that. So if you'd like to go over by yourself, you should Google Baltimore Information Center at Inner Harbor. If you'd like to go over with a walking group, we have two, one leaving from this location at 6:30 p.m. and another leaving from the Marriott at 6:45 p.m. And again, you want to be there by 7. Ship is taking off at 7:30, but really we need people there early because boarding will take a bit. And so with that, unfortunately, Cynthia J. Musanti wasn't able to make it, but luckily we have Ted Rieger here in her stead, and we're looking forward very much to his talk. Ted received his PhD in chemical engineering from Northwestern, and he currently works at Pfizer, where he is a senior principal scientist in quantitative systems pharmacology in early clinical development. So right now, the work that he does supports programs in cardiometabolic, in the cardiometabolic space from early discovery through proof of concept. So with that, thank me in welcoming Ted.
Well, thank you very much to the organizers for the invitation for us to present some of our work today on understanding the role of quantitative systems pharmacology in drug discovery and development. I think this is going to be a little bit of a different talk than we've had earlier today in that there won't be any Julia through most of the talk until the very end. But what I'm hoping to do today is kind of reach out to this community and kind of impress upon you how we're using model-informed drug discovery within Pfizer, and we have certain workflows and certain approaches, and there really is a need for improved tools and improved algorithms to really optimize that workflow and help this reach the next level. And so that's what I'm hoping to accomplish over the next 40 minutes or so. Okay, and again, CJ Musante was very sorry she couldn't be here today, but I'm happy to present her slides on her behalf. So an outline of what I'm going to talk to you about. I'm going to spend a few minutes kind of talking about model-informed drug discovery and development, and that'll involve first starting at the very basics of what is drug discovery and development, and then building up to how do we use modeling and simulation in drug discovery and development, and then we'll start transitioning the types of modeling that we do in my group, which is called Quantitative Systems Pharmacology. So we'll spend most of the talk basically going through QSP 101. We'll talk about what are the types of models that we develop. What are the current workflows that we follow? How do we parameterize these models? How do we kind of explore the uncertainty in these models? And then we'll roll right into showing how we use that workflow as part of a case study to try to deliver value to Pfizer's drug discovery process. And then I'll finish with the last few slides on some of the challenges, opportunities, and future directions that we see for the future of this field. Okay, so the overarching challenge is that we at Pfizer want to bring new medicines as efficiently and quickly as possible to patients in need. If you're familiar at all with drug discovery, you know that it's a long process, it's lengthy, it's costly, there's a very high risk of failure. It begins in the preclinical space. with experiments in test tubes, in cell models, in animal models, and then of the hundreds of compounds, the thousands of compounds that you may test, maybe a dozen of those will reach clinical trials where we start to test experimental medicines in human trials. This is the space that we actually work in. I'm in part of Pfizer's early clinical development team. And if you're familiar with clinical trials, prior to approval, there's essentially 3 phases. In phase 1, you're essentially establishing the safety of your molecule in healthy volunteers, maybe tens to 100 patients. In phase 2, that's where you really start to establish the efficacy of your molecule. And so you're starting to put your molecule into the intent to treat population and looking for signs that you are actually having a positive effect on the disease state. And then finally, phase 3 is what we often call full development, where you're really building your database, building up your safety and your efficacy database for submission to the FDA that this should be a medicine that we want to use for the treatment of these diseases. Now, what you can see as you kind of go along this pipeline, and this is just a cartoon schematic, I'll show you the exact numbers in 2 slides, but you can kind of see this continual necking of potential new medicines, where things are dropping out for many different reasons all along the pipeline, and eventually it takes a lot of effort just to get one approved medicine to market. And so, of course, we're interested in optimizing this process, and one way that Pfizer's really investing in trying to optimize this process is through really enriching our quantitative thinking about our programs, about our molecules, through model-informed drug discovery and development, or what the industry now calls MID3, model-informed drug discovery and development. And it's not new. It has a long history and a well-established role and impact, and it kind of spans across the entire pipeline. Where if we start on the left here, now this is kind of roughly that pipeline again from left to right, you think about kind of systems biology approaches, more network-based approaches, where we might be mapping large genomic databases or expression arrays onto pathway maps to see which are the likely candidates, which targets should we be pursuing for the treatment of a disease. And then as we start honing in on a target, we start developing molecules against those targets. And then that's when our translational sciences groups comes in and starts to think about, are we choosing the right molecule? Are we going to get exposure at the site of action? What happens when it actually binds to that receptor or that enzyme? As things transition into the clinical space, the clinical pharmacology groups have a long history of doing what we call exposure-response relationships, where we're trying to figure out what's the right dose that we should be giving people that's both well-tolerated and likely to be efficacious. And finally, things like precision medicine. Are we testing this in the right patients, or is there special populations that require special handling, such as pediatric populations? There's a lot of different types of modeling that are taking place in drug discovery. Having said all that, our attrition rate remains extremely high. As I said, 10 to 15% of compounds that reach the clinic may eventually see FDA approval. And one particular pinch point that we really are focusing on as a company and as an industry is the area between what we call phase 2 and phase 3. Again, this is when we've just started to test these molecules in kind of a broader patient population, and so we're kind of getting that first set of data about how efficacious these things are. And as you might imagine, given that we're seeing only about a 30% success rate in this space, we've done a lot of studies on why is this remain the highest pinch point. And the greatest reason for failure there between phase 2 and phase 3 is actually efficacy. It's not safety is also another reason. You probably can't read that pie chart, but that's kind of appearing there in red. But in purple is lack of efficacy. It's the first time we've kind of gone from, say, a few weeks of dosing to a few months or even a year of dosing, and it just turned out that these molecules aren't differentiating from the placebo group. And so, we need some other way of thinking about how to get kind of an early picture of how efficacious these drugs are likely to be. Is there any way to kind of get ahead of the game before we do these long, expensive studies, what we call proof of concept studies, and think about, is this the right approach for treating this disease? And we'd like to think that the type of modeling that we tend to do, called Quantitative Systems Pharmacology, might help with this. So, and this is a review article from some of my former colleagues, Paolo Baccini and Piet van de Graaf. And the idea is at the end of phase 2, you declare something called proof of concept, and this sets off certain meetings with the FDA, and you move things into full development. And to really get there, to really have confidence that we're going to have a successful proof of concept, we have to have two things. We have to have confidence that we have the right compound, right? Is it getting to the site of action? Is it engaging with the target of interest? And is it actually doing anything to that target of interest? Is it actually modulating it in some way, inhibiting it or agonizing it? And these different things are usually addressed traditionally through our pharmacokinetics, pharmacodynamics colleagues, our pharmacometrics colleagues in the clinical space. But what we want to kind of add to that through modeling and simulation is some confidence in the target itself, right? Once you bound to that target, how likely are you that you're going to actually move the needle on the disease, that this is actually going to do something downstream that's beneficial for patients? And that's where we think systems pharmacology can kind of come in. Okay? So again, what is QSP, or Quantitative Systems Pharmacology? We build mathematical models and simulate them to mechanistically describe the time course relationships between when we modulate a target and how that propagates through the network downstream to disease biomarkers and the clinical outcomes of interest. Right? And the goal there is to predict and interpret the clinical response and to hopefully improve our decision making as early as possible in the pipeline. That's the kind of 30,000-foot introduction. What I'd like to now do is move into talking a little bit about the QSP workflow and how we actually build and interrogate these models. Okay, so components of a typical QSP workflow. Essentially, a lot of what I do is actually not modeling or mathematical in nature. A lot of what I do is distilling down the biological literature, right? It's sitting down with my biology colleagues, with experts in the field from universities, and really asking, what is the biology that we're studying here? You know, how does everything kind of connect together in sort of a wiring diagram? And I'll show you an example of that on the next slide. Once we've kind of established that biological knowledge, what we think is the relevant biology for our target and our system of interest, then we of course have to convert that into a mathematical model. There's a lot of design decisions that could kind of go into that. I'll touch on that over the next slide or two. But essentially, you know, these models are going to be usually systems of nonlinear ordinary differential equations, where the various species in the biology, the things like glucose and insulin, those physical species are going to be equations in our sets of ordinary differential equations, and then the interaction terms between those different species are going to be parameters. And so a lot of what we do is we have to figure out ways to kind of handle those parameters, through sensitivity analysis, identifiability analysis, estimation of the parameters. We have to somehow parameterize the model so that we can do the simulations. Once we think we have a reasonable representation of the model, then we can go forward into our simulation and predictions, and hopefully we can have some kind of model validation step where usually there is either a piece of data that we hope that we can hold back during the fitting process so that we can test the model, or often what happens is one of our competitors will publish a similar compound through one of their own clinical trials, and that's a good opportunity for us to use the model and kind of confirm that we were able to simulate their compound as well as possible, so that we have some confidence that going downstream we'll be able to simulate our own. And then, of course, I've drawn this not as a linear chain, but as a wheel for a reason, because often what happens is you go through all the steps of the model evaluation and validation, et cetera, and you find that the model's just falling down at some point. And then you kind of have to go back around the wheel again and iterate through the whole process again. Okay. So, what do QSP models usually look like? Well, actually, they usually look like cartoons, bubbles and arrows, like you might see in a review article, right? So we're trying to establish what we think are the most essential features of the biological system of interest. And what I'm drawing here is one of my favorite little models that I've been working with. And what we're looking at here is the details of the biology aren't so important about what I'm going to be talking about for the rest of the talk. But essentially, if you're curious, this is a model of lipid handling in the hepatocytes of our liver. It's meant to simulate a disease state called non-alcoholic fatty liver disease, or NAFLD. Okay? And, you know, what we tried to capture here is kind of the essential species, the essential dynamics, the essential feedbacks that we think are important for when we actually put, for both the base case and we put our drugs on board. We know that we've made a million different design decisions when we kind of drew the schematic. For example, there's no ceramides on this diagram. There are no, you can have a million different combinatoric species of saturated versus unsaturated fatty acids on this diagram. But we've made design decisions to kind of distill those things down because we think that those are kind of secondary to the main effects that we're going to see with our drug. Okay? So that's probably the first part of the art of modeling, of building these models, is the millions of thousands of little different design decisions you have to make, kind of distilling down biology, which sometimes feels infinitely complex, down to something that's kind of manageable for doing your dynamic simulations. Okay? And then the next step, of course, is to build the mathematical model. Now, we always show those cartoons. Biologists like them. They're easy to convey. But behind the scenes, those bubbles and arrows need to become systems of ordinary differential equations. And this is essentially just accounting, right? So all of these little ovals here are different species in my model. So, for example, if we just go around the ring here with triglycerides, I can just do my accounting. Each of these red bubbles here is a flux. That's mass that's kind of crossing into and out of the boundary of certain compartments of the body of the system, and I can just write out my differential equation by kind of going around the ring and seeing all the inputs and outputs of each of these compartments. So if I look at the change in liver triglycerides versus time, this will be 1 OD in my model. It's equal to the hepatic TG uptake rate plus the rate of esterification, if you can see that little red arrow on the left of that pool, minus the outflow due to lipolysis. Okay? So that's all just accounting. But now the second art of modeling comes in. It's deciding when to make assumptions about linear kinetics versus more hyperbolic kinetics. What mathematical form are each of these fluxes going to take? And so that's also a lot of the design decisions that you have to kind of put in the model, because, you know, a lot of different mathematical forms might fit the model just fine at baseline. When you just kind of simulate the model to say steady state, there's probably a lot of different ways you can write these equations and be just fine. But it's when you kind of push the system, when you put on board therapies and start pushing a lot of flux through the model, that really those kinds of assumptions will break down very quickly. Excuse me. Okay. So we need to use some data to kind of inform those mathematical structures and to start to parameterize the models. People always ask me what types of data do we use and how am I big data? I actually wish that I was bigger data than I am. It turns out that when you're kind of working in the clinical space, especially in diseases like NAFLD, which have no approved therapies, so there's not a lot of clinical data out there yet. There's not a lot of data out there for us to be using. And we focus very much on the human data because that's obviously the most relevant to what's going to happen in our clinical trials. And we look for data like what I'm showing here. This is a clinical trial where essentially they were adding tracer studies and looking at the rate of VLDL, very low density lipoprotein export from the liver. Essentially, when you go to the doctor's office and you have you're in your physical, they take your cholesterol, they take your triglycerides. This is a big component of that triglyceride pool that they report back to you, and it originates from your liver, right? And what they saw was a fairly, each of these circles here and triangles here are a different patient, or one snapshot of a patient. And what they saw was a very nice hyperbolic relationship between the rate of secretion of those triglycerides and your intrahepatic fat content. And so that's a very nice way to kind of establish that relationship within the model directly. And so what we're usually looking to do is to kind of take that data, and we're not usually looking to simulate individual patients, and there's a lot of different reasons for that. I'll touch on that on the next slide. But we're usually interested in kind of figuring out what's going to be what's kind of the population distribution of some of these parameters, and what's going to be the likely population level response to a drug. So we can just kind of take that data from the previous slide. We can do any kind of nonlinear regression on it, fit a kind of a hyperbolic curve to it, and come up with some estimation of the mean and the confidence intervals or prediction intervals of that data, and those would be parameters and ranges that you could use directly in your modeling. On top of that, so that would be parameters for species in the model. You know, we do have generally population level data for certain sets of the observables. For example, if I was interested in simulating cholesterol metabolism, well, there is very good studies out there. In this case, we're showing the NHANES data from the NIH. And what they did was they did a survey of the distribution of these parameters in the general American population. So what I'm plotting here on the top row is HDL, LDL, and total cholesterol. Again, you go to the doctor, you get a physical, they measure your cholesterol. That's good cholesterol, bad cholesterol, and total cholesterol. And kind of showing the joint distributions down here at the bottom and how these three things are kind of related or correlated. And so this is kind of forming the constraints for what's plausible for certain species in the model. So we've constrained the parameters, and now we've constrained the species at kind of a population level. Now, why do I keep talking about population level and not maybe, how do we simulate individual patients for precision medicine? Well, I think the fundamental problem is that these models are rarely identifiable. QSP models come in all different flavors, from the very large systems, huge whole body models of metabolism that I used to work on at my previous job at Entelos, where we were dealing with 800-900 ODEs and thousands of parameters. But they can go down in size to more of these more of the kinds of mechanistic models that I tend to build more nowadays at Pfizer, where it's more of a model of a pathway or a tissue, much more focused. But even there, if you have 10, 15 species, there's no way that enough data has been collected on each individual patient that you can identify the parameters of the model. In fact, it's not until you start getting down to the very smallest models, maybe 3 or 4 equations. What I'm showing on the very right is the famous example is the Bergman minimal model, where if you correctly measure insulin and glucose in a set of patients, you can actually back out from that model their insulin sensitivity and beta cell function of the patients. But those are essentially glorified regression models. For practical reasons, these models are never identifiable. So we need some way of kind of simulating the population level response and exploring that parametric uncertainty. So what we usually do is we usually simulate not just one mean fit, not just one best fit of the model to the data that's available, but we usually try to simulate ensembles of alternative parameter sets. And what you can imagine is I've just shown you how we can establish ranges for some of the parameters. I can draw as many of those things as I want, whether they're correlated or uncorrelated, but I can just keep drawing them tens of times, hundreds of times, thousands of times, hundreds of thousands of times, and I can build up a nice set of parameters that I can then run through the model. And I can see how those things kind of map onto, say, my observable space. That is, the things that I want to observe in the clinic. This could be glucose and insulin, liver fat and plasma cholesterol, whatever. And then what can be really interesting is, because it's a nonlinear model with plenty of feedback in it, even if things look the same at baseline, for example, in the purple and the blue here, might look very close at in terms of baseline, they might have differential responses to therapies. And that's something that we would like to identify and try to learn more about using the modeling. And so that's, again, another reason why just kind of optimizing to a single point, a single value of glucose at baseline wouldn't be adequate. You have to really have explored all the uncertainty in your parameter space. The other thing we want to keep in mind when I talk about population level responses is that it's not just about filling the space. It's not about having as many simulations as possible. We want to be a lot smarter about it. For example, if I ran it many, many more times and I kind of populated out that space from the previous slide, it might look like what we have on the left here, where we've kind of space filled this two-dimensional graph. But imagine now your likely clinical population actually falls within this ellipsoid. So these people that are kind of these circles, these simulations of the model that kind of fall outside of this ellipsoid, I can't exclude necessarily that these people exist, that there might be one of these people on the Earth, but they are extremely unlikely to ever walk into one of our clinical trials. And so we would like a way to maybe downweight those solutions which kind of fall outside of that distribution, and to really focus on the most likely patients that we're going to be treating with our drugs. Okay, and so about a year ago we published this as kind of a commentary, and I think that all of our kind of sister groups that are competitors, they follow not the same exact one, but a very similar style workflow, which is, we called it the virtual patient and population workflow. I've kind of taken you through most of the steps already. You implement the model, you define those plausible ranges. Well, how wide, what's a reasonable range for the parameters of the model? What's a reasonable range for all the physiological outcomes of the model? A cholesterol value of between 30 and 400 might be acceptable, but probably a cholesterol value of 10,000 is not. Once you've kind of defined all those plausible ranges, you can then use your favorite global optimization algorithm, whatever that is, genetic algorithm, simulated annealing, Metropolis Hastings. We showed Metropolis Hastings algorithm was actually fairly efficient at kind of exploring the space for one type of model, and try to populate out that space as much as possible. You can then take the final step in the process is then you can take the model through and all those solutions to the model through acceptance rejection sampling and essentially come up with a virtual population that hopefully matches all your predefined criteria and also reflects the statistics of the population of interest. So just to close the loop there and kind of show what that acceptance rejection sampling looks like. So I showed that cholesterol data a few slides ago. This is that again, the NHANES data, beautiful log normal distributions. The black there is the NHANES data, the distributions. The red is on the top row is on the left is the distribution of our plausible patients, all the simulations from a model, and then on the bottom row is the individual points. You can see that everywhere where there's black, there is some solutions of the model, which is good news. But you can see we've wasted a lot of time, and a lot of meaningless solutions have been generated that kind of fall outside of those ranges. And so the final step there was to kind of take it through acceptance rejection sampling and come up with a population that met all those preset criteria and also reflects the distributions of the population. We can then take this virtual population, forward into studying a novel therapy for cholesterol metabolism, for example, a new PCSK9 inhibitor. Okay. So I've walked you through the workflow. I've kind of introduced the problems. I've walked you through the workflow. What I'd like to finish with is a case study and how we kind of use that whole workflow to hopefully bring some value to Pfizer's drug discovery process. This is not an example from my work. This is from my colleague Richard Allen, who's currently attending SMB, so he couldn't be here today. But the idea was that we wanted to use our modeling simulation to get a better handle on what's happening with a ketohexokinase inhibitor. This was a novel treatment for non-alcoholic fatty liver disease, and at the time that I'm kind of telling this case study on, the drug program was in phase 1 of development. It had already been through kind of those initial trials in patients. We had some data in humans, but we hadn't yet run those large trials to really get a good handle on efficacy yet. And the question came back from the team is, what's the predicted level of target engagement based on changes in plasma biomarkers, right? So we have a target that's in the liver, but all our measurements in phase 1 are going to be in the plasma, in our blood. And while it's possible to take direct biopsies of the liver, it's not something we're going to do very early in development. It's uncomfortable, it's expensive, and it can actually be dangerous for patients. So it's something we'd like to do carefully, judiciously, only when necessary. So, but could we use the model to try to infer what happened in the liver based on our observations in the plasma? And that was the idea. Okay. I'm not going to, again, the details of the biology are not so important, but ketohexokinase, if you're curious, and I'd be happy to talk a lot more about the biology later today on the cruise, whenever. But essentially, we all have fructose in our diet. Ketohexokinase catalyzes the transformation of fructose to fructose 1-phosphate, which is the first step of its metabolism in the liver. Fructose has been metabolism of excess fructose has been implicated in non-alcoholic fatty liver disease, and so this was thought to be perhaps a fruitful path to treating the disease. Okay, what the clinical team was thinking about this in the model is what's shown in the upper right there. Again, the details are not so important. Another bubble and arrow diagram. But what the clinical team was thinking about doing was they were going to run an imaging study where essentially they were going to put the inhibitor on board and then try to image the liver and try to get a picture of what's going on in there to see how much target engagement we might be getting. The problem is that there was going to be a long lead time to build the reagents for this. It was going to be an extremely expensive study, and even if all those things had panned out, we weren't confident that the methodology was going to give us back the answer that we wanted. Not that we wanted, that was going to answer the research question adequately. And so the idea was, well, why put patients through this unnecessarily when we can use the model to try to make the same calculation as we had before? Okay? So what Richard did was he kind of followed the workflow that I've been outlining here. He had a fructose model with a virtual population that was constrained based on a certain set of literature data, right? So he built the model without looking at the response of our inhibitor in the clinic. He then brought in that novel data together with the virtual population that he already made, and kind of took a look at if there's a subset of virtual patients that match both the literature data and the data we saw in a clinical trial. So essentially, he's going to take this big hypercube of observations, and he's going to slice through it and say, okay, this subset of observations, this subset of simulations actually is consistent with all of the data. So that's going to be my new virtual population. And then I'm going to use that virtual population to I'm going to put the inhibitor on board to different strengths and look at the change in the plasma biomarker. That's plasma fructose. And I'm going to ask at what level of target engagement were we likely achieving to get those observations in plasma fructose. So working our way backwards from the endpoint to the input. And then we can make a program decision based on that analysis. So there was obviously a lot of uncertainty. There's uncertainty in the parameters of the model. There's clinical variability, and then on top of that, there was uncertainty in the level of penetrance that our molecule was getting into the liver. Our DMPK scientists, the scientists that are in charge of studying the distribution of our compounds in the body, they gave us a range. It was somewhere between a value of 1 and 10. So what Richard had to do is he kind of rolled that into his simulations in terms of uncertainty, and what he was able to show is that, despite all this uncertainty, despite a range between a KPUU, which is just the partition coefficient between the plasma and the liver, of either 1 or 10, none of that mattered. We had gone sufficiently high in our dosing in phase 1, in that we could have reasonable confidence that we had fully engaged this mechanism in the liver, and we didn't have to test higher doses going forward. So what's the answer? Kind of wrapping up the case study. The systems model predicted we had robustly modulated the target already. We didn't need to use higher dosing, and we could rest assured that we had kind of achieved what we call proof of mechanism. We had seen some early signs of efficacy, that we were actually able to engage the target and get a meaningful biomarker movement in our study. And what this was used for is we were able to eliminate that imaging study, saving Pfizer a few $1 million in a few years, in a year probably of development time. Okay, so that's kind of the end of the case study. What I wanted to finish up with is just a kind of a few comments on where the field is and where it might be going in the next few years. Okay. So I thought I'd bring it back a little bit to tools. So this is a survey done of QSP software tools by one of my colleagues who's now at Amgen, Sergei Ermakov, where he kind of surveyed the field. He got a broad cross-section of researchers at different companies, different institutions. What are the software tools that they're presently using for this kind of modeling? And what you can see here, especially if you combine MATLAB with Symbiology, is certainly the MathWorks products remain dominant in the field. Secondarily is R and its various packages. Certainly, especially in people coming from more the pharmacometric space, R is deeply penetrant in that workflow. And the final one is that's kind of common, and you can see there's a half dozen other ones which are a little less common, but non-MEM, because a lot of what we do is, since we're interested in population level responses, is nonlinear mixed effects modeling on our data. And so non-MEM is kind of a specialized tool in Fortran for doing that type of modeling. What's interesting at the bottom here is the secondary question was, what are you happy with about your software? Where does it excel? It looks like a lot of people are actually happy with the tools they have for building the model. But running simulations, visualizing the results, and especially parameter estimation and data fitting seems like a good place where the field feels like their current software is kind of falling down. So, why are we here today? What are we excited about? What new tools and approaches are we looking forward to? I think better computational tools will open up exciting new avenues for leveraging modeling simulation across drug discovery and development. Especially faster simulations will allow us richer timescales and really put more mechanistic into mechanistic modeling. Our models will always be practically unidentifiable. It's just a reality of the types of modeling that we do. But faster and more parallel simulations may allow for more complete exploration of that space, so that we have more confidence that we've kind of gone through the space a little bit. And we haven't talked about this at all, but really integration with machine learning approaches is kind of a hot area, and it's something that we really need to think about how we can connect the kind of continuous models that we build that maybe lead to certain biomarker changes to certain outcome measurements that we're really interested in the clinic, for example, resolution of a disease or improvement in an ordinal score of some type, like the NAS score. And so, things that kind of get us excited about Julia is we hope, and we currently are doing a kind of a pilot project with Julia Computing to try to translate some of the workflow into Julia, and I hope to have a report on that next year. But I think it might help us address all of these things in something. And one thing I actually barely touched on in this presentation, but it's really a bottleneck in our workflow, is we spend a lot of time translating our models from the scripting language, from MATLAB to C, for speed, just for performance reasons. And that's a real bottleneck, especially when you remember that our workflow is a wheel, and it makes iterating painful. Okay, so hurdles. Institutional user inertia is just probably the biggest one. We've spent a lot of capital and people time invested into R and non-MEM, and that's just the reality. I haven't touched on the regulatory aspects of what we do, but there is a regulatory compliant environment in place for R and non-MEM at Pfizer, and so that's another hurdle that would have to be overcome. And just want to touch on this briefly. Just kind of, we're end users, we're not developers. And so we do rely on there being high quality documentation and some form of stability in the packages that we use. And so that's another consideration that I want to kind of learn more about over the next few days. Okay, so I think I'll skip the summary because I know I'm running out of time. So I just want to kind of put up my acknowledgement slide. Obviously, I want to thank CJ. She's really sorry she couldn't be here today. Richard Allen, who did a lot of this work with me. And I also want to thank our leadership team, Jeff Pfefferkorn, John Lucanucci, for having a lot of belief in this group and what it does, and letting us kind of explore this project with Julia Computing on can we make these things more efficient. Okay, and with that, I'll close. Thank you all very much.
So one of the things that I was a little surprised to see was the absence of a return to animal models or things like that once you've built these, sort of, let's say, your first quantitative networks. Is that really something that isn't practically useful, or is that just something that you took for granted happens all the time?
So, I'm not sure I entirely understand the question.
Well, meaning, so I'm guessing that in many cases when you're building a model, that model has never previously been built or tested in a direct, concrete way. And I mean, one of the things I'm imagining, right, is if you insert an extra feedback loop or missing an important feedback loop somewhere in the system, it could pretty substantially change the dynamics of the system, right? And so that's where I'm wondering where if this is really the first time you've put together a whole system view of it, does it make sense to go back to a cheaper system for a period of the exploration?
Yeah, so certainly if the model is making an important prediction, right, about safety or just that this approach is not going to work, right? So certainly that would be the opportunity to engage with our preclinical colleagues, where we could go back to the animal testing and maybe design, the model has made a specific prediction, has made a specific hypothesis that we can now test in a rodent species and see, is this a real concern or not? Unfortunately, mice are not humans, so it's not always a perfect prediction, which is why, we've certainly done a lot of animal testing that hasn't held up in the clinic, and so that's why there needs to be more clinical, more human-focused modeling done.
There's the analogy with the pipeline. I was just wondering, how would you say the pipeline was built? I mean, where does all this knowledge come from? All
the biological knowledge?
Yeah, I mean, like all along the pipeline, it's where does it come? Where do all these things come from? So, I mean, how does it fit into, what is this business model actually?
What is the business model?
Yeah, I mean, in terms of the pipeline.
So we're a drug development company. I'm not sure.
Yeah, but if you look at like all the things and that makes up the pipeline. I mean, well, I'm just.
So are you asking why we do basic research?
Yeah.
No, that's fair actually. Some companies have don't see the value there. In doing basic research or even early phase clinical development, and essentially they would just become an in-licensing company. But essentially, that comes down to an economics argument of are you getting return on your investment in R&D, right? Are you covering cost of capital that you invest in the R&D space? Because if you start to try to always in-license late stage assets, you're paying an extreme premium to get them in-house, and you still might have only a 40% chance of them reaching market. Does that answer your question?
Yeah, and I think we have like very powerful scientific models, but we still know very little biologically. I think, at least.
That's what makes this job so interesting. Yeah.
Okay, so this on this chart here. So at what point, I assume this represents let's say, all ideas on all diseases that have been thought over the past 2 years, and they're all just kind of traveling through this funnel. So at what point do you get to where you have a specific disease, and let's say you have an idea of approximately the molecule to treat it, but you have 14 different versions of the molecule that are all very, very slightly different, so at what point do you go from 14 to 1, or is that different for each case?
So there's a very standard process that we follow. So you start off with, we have certain research areas that we're interested in. For example, cardiometabolic disease is the one I'm affiliated with. So that's how we choose diseases, right? And then once we've kind of identified a target, however we did that, whether it's from thin air, from the literature, from analysis of large genetics databases, which is more and more how we do this, we choose a target. We can then do screening. But Pfizer and all of its competitors maintain libraries of millions and millions of compounds that we can then push against a specific target in a high throughput manner. From that, we'll get hundreds of hits, which can then be tested experimentally, and they'll kind of winnow the way down until we finally have one candidate molecule, which is nominated for doing all the regulatory toxicology experiments, making sure that these things aren't going to be carcinogenic, et cetera, that are required to actually put this compound into humans. So there's a long process going on here that can span anywhere from 3 years to probably 7 years in the green boxes that are on the far left there.
So I come from a background in modeling and simulation, and we've done some work with machine learning. And one thing that we've done is hooking up your modeling and simulation to your machine learning results to suggest more modeling and simulation, so you can guess in the right. You can feed the machine learning where it needs more information. So the machine learning can inform the modeling and simulation pipeline where it needs more data. Have you guys considered anything like that?
Probably not in such a formal way, but certainly, through just kind of interrogating the models, and doing global sensitivity analysis, doing local sensitivity analysis, you do kind of get a picture of where more data would be most beneficial for helping to identify the models better.
So you might want to consider hooking that up hard so it's actually in the loop.
I think I'd love to talk to you some more. Okay. Do we want to take one more, or are we all done? Okay. Last one, I guess.
Okay, thanks. I was wondering, most of the time that you talked about, like, whatever molecules you guys were studying, it was sort of one molecule for one disease and not tandem therapies. I wonder, do the modeling pipelines that you guys use currently take into account, like, multiple drugs being used at the same time for one thing, and how does it deal with the combinatorial explosion?
So a lot of what we actually use these models for is considering the impact of combinations of therapies, because a lot of times when people have very serious diseases or very chronic diseases such as type 2 diabetes, you're not just on one therapy. You first you go on one, then you go on a second, then even a third line therapy, and so we have to consider all of those background medications in terms of its impact on the biological system, and how adding on top a new therapy might impact both safety and efficacy.
And with that, let's thank our speaker.